For your next video, let's focus on the structure and function of the plasma membrane. As you'll read in detail in the paper presented this week, the fluid mosaic model for the plasma membrane was presented to the scientific community in 1972. The plasma membrane of the cell is kind of a mosaic mix of phospholipids, cholesterol, proteins, and carbohydrates. The membrane ranges from about 10 to 12 nanometers in thickness and appears to have a sandwich-like structure with toothpick proteins dotted right through it. Those carbohydrates, it's important to note, are only present on the exterior surface. Some of them are attached to proteins, and we call them glycoproteins, while others are attached to lipids, and those ones we call glycolipids. We're going to look at all these little things more closely in turn. Phospholipids make up the major components of the plasma membrane, and they have a hydrophilic or water-loving head and a hydrophobic or water-fearing tail. A phospholipid molecule contains a 3-carbon glycerol backbone with 2 fatty acid molecules that are attached to carbons 1 and 2, and then there's a phosphate-containing group attached to the 3rd carbon. The structure is seen here in both the chemical stick model and then a really basic cartoony version down there on the bottom. In an aqueous solution, phospholipids tend to arrange themselves with their polar heads facing outwards and then their hydrophobic tails facing inwards. You can see here in these different depictions the kind of structures phospholipids tend to make entirely on their own. Proteins make up the second major component of plasma membranes. Integral proteins get their name from the fact that they are integrated into the structure. They have amino acid regions that are hydrophobic, and this keeps them anchored within that middle section of the plasma membrane where their chemistry mixes well with the hydrophobic tails. Those little bits are about 20 to 25 amino acids. Why? That's how much it takes to span the width of the plasma membrane generally. There can be just a single pass of a hydrophobic protein, or there can be many passes of hydrophobic protein, and they can actually create their own little channels. The hydrophobic sections can be the alpha helice in shape, or they can be a beta-pleated sheet. Either is just fine. Peripheral proteins, though, those proteins are a little bit different. They don't do this membrane spanning thing. They are actually found on the exterior and interior surfaces of the membranes. A lot of those peripheral proteins, they act as enzymes, so they help chemical reactions take place, or they are considered cell recognition sites. So they kind of function like fingerprints. The third major component I mentioned are carbohydrates. Carbohydrates are often found on the exterior sides of cells. They can be as short as just 2 little monosaccharides together, like sucrose seen here, or as long as 60. These largely exist as identification molecules, which is super important to the immune system. Carbohydrates are very hydrophilic, which is helpful because they actually draw water to the surface of the cell, which allows the cell to maintain its health. Carbohydrates on the outside of cells lead to some pretty interesting things in medicine. Viruses actually infect specific organs or specific cell types by recognizing the glycoproteins and glycolipids that are hanging off the outside of the cells. The viruses have little receptors that look for those specific glycolipids and glycoproteins. They actually bond with them and dock with them, and that helps them to get a good handle on the cell that they are wanting to infect to inject their genetic material. Not having the correct markers on the outside of the cell can actually save an individual from being infected with certain viruses. For instance, there are some individuals that do not have a particular protein hanging off the outside of their cells, so those people are actually immune to the virus HIV. They're not capable of being injected with the genetic material, so while they can get little particles of the virus, it will not proliferate in their system. We're going to look at that more closely. So here are some components and functions of the plasma membrane if you want to jot them down. This table is found in your book. So it's important to note that those membranes are asymmetric. The inside doesn't match the outside. On the interior, there are proteins that serve as anchors, and they are actually attached to the cytoskeleton. On the outside, some of those proteins serve as anchors, but they have attachments to the extracellular matrix. The amphiphilic nature of the membrane allows some molecules to easily move through it at will, like little guys, oxygen, carbon dioxide, vitamins, lipids. Other substances have to move through specialized protein, though, and therefore our cells can closely control the movement of these large molecules super specifically, and that helps keep our cells nice and healthy. So about that easy movement, the easy movement is called diffusion, and it's known as a type of passive transport. Substances move naturally from high to low concentration, and that's just a property of physics. You can see a simplistic cartoon of diffusion here, and there are multiple factors that affect diffusion, which I'll show you on the next slide. So the 7 factors that affect diffusion are the concentration gradient, the mass of the molecules, the temperature, the solvent density, solubility, surface area and thickness, and the distance traveled. I don't expect you to memorize that there are 7 factors that affect diffusion. You should know, however, how each one of these things does affect diffusion. I'll leave you to write this down. Not all transport is passive. Some transport must be facilitated. There has to be help to move things across the plasma membrane. Most of that movement is assisted by membrane proteins. The things that must move through membrane proteins are things that are polar. They chemically don't get along with that plasma membrane because they're repelled from either the outside of the plasma membrane or the middle of the plasma membrane. They can never pass through completely on their own. Membrane proteins can either be channels or carrier proteins, and we're going to look at both. The integral proteins that are involved in facilitated transport are collectively referred to as transport proteins. Let's take a look at those 2 specific types. Channel proteins have hydrophilic domains exposed to the intracellular and extracellular fluids, and additionally, they have a hydrophilic channel through their core, which creates a hydrated opening through the membrane layers. Aquaporins are a special type of channel proteins that allow water to pass through this membrane at a very high rate. Channel proteins can either be open at all times or some of them are gated, so they have various mechanisms to control their opening. The attachment of an ion to channel proteins may control that opening, or the mechanisms may be some other substance involved. In some tissues, sodium and chloride pass freely through open channels, where in other tissues, those same exact 2 chemicals must pass through a gated opening to get into and out of the cell. Another type of protein embedded in the plasma membrane is called a carrier protein. This aptly named protein binds to a substance and in doing so changes its own shape. This moves the bound molecule from the outside of the cell to the inside of the cell. Depending on the gradient, the material may move in the opposite direction. Carrier proteins are typically specific for a single substance. Channel and carrier proteins tend to transport material at very different rates. Channel proteins transport things much more quickly than do carrier proteins because you don't have to wait for that change in shape. Channel proteins facilitate diffusion at a rate of tens of millions of molecules per second, whereas these carrier guys seen here, they work more like 1,000 to 1,000,000 molecules per second. Osmosis is the movement of water through a semi-permeable membrane according to the concentration gradient. Water moves from an area of high concentration to one of low concentration. This occurs when a solute cannot move, but the water itself is capable of movement. Osmosis can be measured essentially by a property called tonicity. What tonicity refers to is the fact that extracellular solutions can change the volume of a cell by affecting its rates of osmosis. Tonicity is correlated with the osmolarity of a solution. So let's unpack some of those words. Osmolarity is a reference to how much solute there is. Osmolarity is measured by something called tonicity. Tonicity is the tendency for extracellular solutions to change the volume of a cell by affecting the rates of osmosis. And this is all correlated with the osmolarity of a solution. Osmolarity is the total solute concentration of a solution. Solutions that have extremely high osmolarity rates are going to affect the way that water moves. If you have more solutes, water is going to move towards that solute much more quickly than if you have a lower solute concentration. We refer to the tonicity of solutions 3 different ways. Hypotonic solutions are solutions where the extracellular fluid has lower solute levels than those found inside the cells. In hypertonic solutions, extracellular fluids have a very high solute concentration level compared to that of the cell. In isotonic solutions, osmolarity of the extracellular fluid is almost completely equal to that inside and outside of the cell. To put this concept into real-world terms, our blood has a specific osmolarity or solute concentration. Our blood cells are approximately equal in solute concentration to the extracellular fluid that exists in our bodies. When a human is injected with a hypotonic solution in large quantities, Our cells will take in too much liquid in an attempt to balance out the osmolarity, or solute concentration, and this causes the cell to burst. The chemical system is trying to lower the solute concentration inside of our cells to match that of the environment they're currently sitting in. Hypotonic solutions make cells look like O's and they burst. If a hypertonic solution is given, the cells will lose water in an attempt to equalize the osmolarity. The cells will shrivel up, collapse on themselves, and become ineffective and die. Both are deadly conditions, which is why individuals who are dehydrated are given an isotonic saline solution. That might sound familiar if you've ever watched any medical shows or even fake ones like Grey's Anatomy or House. Some organisms, such as plants, fungi, bacteria, and some protists, have cell walls that surround the plasma membrane and prevent that lysing or exploding in the hypotonic solutions. The plasma membrane can only expand to fit the limit of the cell wall, so the cell can't lyse. In fact, the cytoplasm of plants is actually slightly hypertonic to the cellular environment, and the water will always enter the cell if available. This inflow of water produces turgor pressure, which stiffens out the cells of the plant. As you can see, though, it's still obviously dangerous for the cells to be hypertonic. There's not much in those cells, so they can't do a whole lot to assist the plant in growing. In non-woody plants, that turgor support, having enough water in your cells, helps to support the plant. Conversely, if a plant is not watered, the extracellular fluid will become hypertonic, causing water to leave the plant cell. In this condition, the cell does not shrink because the cell wall is not flexible. However, the cell membrane will actually detach from the wall and it will constrict the cytoplasm. This process is called plasmolysis. Plants will lose turgor pressure, and that will cause the plant to wilt, as seen on the left. If you water it, you regain that turgor pressure, and you have a plant like the one on the right. Tonicity is a concern for all living things. For example, paramecia and amoebas, which are both protists that we will get to know quite well, they lack cell walls, but they have contractile vacuoles. This vesicle collects excess water from the cell and pumps it out. It keeps the cell from lysing as it takes on water naturally from its environment. While pumping water out might take energy, most movement of water doesn't take much. Active transport is a different type of movement into and out of a cell, and it does take quite a bit of energy. That's where we're headed next. Active transport requires the use of cells' energy because it's often moving things against their concentration gradient. Movement in active transport often occurs via an electrochemical gradient, through primary active transport, or maybe even secondary active transport. The mechanisms that help with this active transport are known as cellular pumps. Ions move into and out of the cells because cells contain a lot of proteins. Proteins don't move across the membrane, and they also tend to be mostly negatively charged. So this creates an electrochemical gradient, a difference of charge, across the plasma membrane. The interior of living cells is electrically negative with respect to the extracellular fluid in which they are bathed. At the same time, cells often have a higher concentration of potassium and a lower concentration of sodium than they do in the extracellular fluid. It's important to note that both potassium and sodium are positive ions. So in a living cell, the concentration gradient of sodium, Na+, tends to drive it into the cell. The electrical gradient of sodium also tends to drive it inward into the negatively charged interior. The situation is more complex, however, for other elements such as potassium. The electrical gradient of a K+ ion also tends to drive it into the cell, but the concentration gradient of K+ tends to drive it out of the cell. The combined gradient of concentration and electrical charge that affects an ion is called the electrochemical gradient. There's a great balancing act in between how much of this ion do I have and how much of this charge do I have. Both are regularly balanced throughout all of our cells all the time. An important membrane adaptation for active transport is the presence of specific carrier proteins or pumps that facilitate movement. A uniporter pump carries one molecule or ion in one direction. A symporter carries two different ions or molecules both in the same direction. An antiporter carries two molecules or ions, but they travel in different directions. Primary and active transport can work together to move ions across a membrane, creating an electrochemical gradient that can be used for various cellular processes. One of the most important pumps in active transport processes is called the sodium-potassium pump. There are 6 steps in this process, and it explains what's going on in this picture here. I'm going to tell you what's happening in this process so you can visualize it as you look at the picture, and then I'm going to show you the text of exactly what I said. So try to just listen and visualize, and then you'll see the text, so you'll have an opportunity to write it all down. With the enzyme oriented towards the interior of the cell, the carrier has a high affinity for sodium ions. 3 ions will bind to the protein. ATP is hydrolyzed by the protein carrier and a low-energy phosphate group attaches to it. As a result, the carrier changes shape and reorients itself towards the exterior of the membrane. The protein's affinity for sodium decreases, and the 3 sodium ions are going to leave the carrier. The shape change increases the carrier's affinity for potassium ions, and 2 such ions will then attach to the protein. Subsequently, that low-energy phosphate group is going to detach itself from the carrier. With the phosphate group removed and the potassium ions attached, the carrier will reposition itself towards the interior of the cell. The protein carrier with its new configuration has a decreased affinity for potassium, and those 2 ions are released into the cytoplasm. The protein now has a high affinity for sodium ions again, so the process starts all over. Some transport or export must occur in bulk, and that's referred to as endocytosis as a general term. There are a few different specific types of endocytosis. Phagocytosis, seen here, is the process in which large particles of solid materials are taken into the cell. A section of cell preparing to pinch in on itself is coated with a molecule called catherine. This increases the stability of the vacuole. Eventually, the entire vesicle will pinch inward, where it migrates and merges with the lysosome. The material inside of it will be digested, and the macromolecules will be shipped to wherever they are needed. Pinocytosis is another type of endocytosis, but it's a little bit different. This is where the cells undergoing the endocytic process bring in fluids and free ions. There's no merging of a lysosome in this process because there's nothing to dissolve. A variation of this process occurs with even smaller vacuoles that are simply passed through the cell and released off to the other side. This is known as podocytosis, and it uses a protein called caveolin to mark these special vesicles. Sometimes we just need to transport things through our bodies, and membranes can help us do that. A targeted variation of endocytosis employs receptor proteins in the plasma membrane that have a specific binding affinity for specific substances. Catherine-coated pits are used in this process too. Receptors pick up the molecules that they need from the extracellular fluid, and that signals the cell that that particular pit needs to be folded in, and then the ions are simply taken to wherever they are needed. It's a very convenient process. The reverse of this process is moving material out of the cell via a process called exocytosis. Exocytosis is the opposite of the process discussed above, and its purpose is to expel material from the cell into the extracellular fluid. Waste material is enveloped in a membrane and fuses with the interior of the plasma membrane. This fusion opens up the membranous envelope to the exterior of the cell, and the waste material is simply expelled into the extracellular space. Other examples of cells releasing molecules via exocytosis include the secretion of proteins into the extracellular matrix, or maybe the secretion of neurotransmitters into the synaptic cleft by synaptic vesicles. These are how our neuronal cells or our brain cells talk to one another. Let's have one last overview of transport energy requirements and the types of materials that can be transported. This graph is a nice wrap-up of all of those methods covered in this chapter. It also ends the last section in your book. We'll end our video on the plasma membrane and transport into and out of cells. As always, read your book, answer the questions at the end of each section, and check Learner for your homework assignments.